Welcome students. In this part, we will discuss fourth colligative property, osmotic pressure, in which we will discuss osmosis, demonstration of osmosis, osmotic pressure, biological significance of osmosis, and reverse osmosis. Osmosis and osmotic pressure. The phenomena of osmosis was first studied by Abe Nolet in 1748. The phenomena of the flow of solvent through a semi-permeable membrane from pure solvent to the solution is called osmosis. It means in osmosis there is a flow of solvent molecules through a membrane, and that membrane is known as semi-permeable membrane from pure solvent to the solution site. Osmosis is a very common phenomena which we observe in nature or at home. For example, raw mangoes shrivel when pickled in brine. Brine is a solution of salt and water. Wilted flowers revive when placed in fresh water, and blood cells collapse when suspended in saline water. If we look into these processes, we find one common in all, that is, all these substances are bounded by membranes which occur in nature in plants and animals. It means the substances like raw mangoes, wilted flowers, blood cells, they all are bounded by a membrane, and these membrane can be found in nature, plants and in animals. These membranes appear to be continuous sheets or films, yet they contain a network of submicroscopic holes or pores. These membranes which is present in these substances have a continuous sheet like structure, and in their network there is a holes or the pores are present, and small solvent molecules like water can pass through these holes, but the passage of bigger molecules like solute is hindered. It means the holes which is present in in membranes can only allow the movement of small particles or small solvent molecules. They hinder the movement of bigger molecules of solute. So these membranes, the membranes which having this kind of properties are known as semi-permeable membrane, can be denoted by SPM. Osmosis can also take place between the solutions of different concentration. If we have the solutions of different concentration, then osmosis can also take place in that cases. In such cases, the solvent molecules move from the solution of low solute concentration to the higher solute concentration. It means when we have two solutions of different concentration, then the movement or the flow of solvent molecule to the solution site will always takes from lower solute concentration to higher solute concentration. Demonstration of osmosis. First of all, take two eggs, remove the outer hard shell of the eggs by dissolving in dilute HCl. Place one egg in distilled water and the other egg in saturated solution of sodium chloride. This can be seen by the figure. If we place one egg in distilled water and other egg to the saturated solution of sodium chloride, what we will observe that the egg which we have put it in distilled water, it will get swell. Why? it swells up. Why? Because due to osmosis, the flow of solvent molecules towards the solution will take place. Since we take distilled water, and in distilled water it is a pure solvent, there is the presence of only the water, no other particles, no other molecules are present in this water, so it is a pure solvent. And in osmosis, the flow of solvent particles towards the solution takes place. So it means this solvent molecule of distilled water will move towards the egg side, towards the solution side, and with the help of semi-permeable membrane, these particles can easily move towards the egg, and the egg will get swell up. Now, in the case when the egg is placed in a concentrated salt solution or a sugar solution, as we have taken the concentrated sodium chloride solution, then in this condition the solution has more amount of solute, since it is a concentrated solution. And as we are talking about concentrated solution, so solution has more amount of solute, and we place a egg in that solution. So what will happen? The flow of solvent will take place from low solute concentration, which is present in egg, towards the higher solute concentration, which is present in the solution of sodium chloride. So the egg will get shrink in this condition. क्योंकि इसमें क्या है, जो सॉल्यूशन हमने लिया हुआ है वो सॉल्यूशन कैसा है, सैचुरेटेड सॉल्यूशन है, मैंने उसमें सोडियम क्लोराइड का अमाउंट ज़्यादा है, मतलब सॉल्यूट का अमाउंट ज़्यादा है। जो सॉल्यूशन हमने लिया हुआ है उस सॉल्यूशन में हमने एग को पुट करा, तो एग के अंदर जो सॉल्यूट का कॉन्सेंट्रेशन था वो लो था एज कम्पेयर टू जो सॉल्यूशन में हम उसे पुट कर रहे हैं. What will happen? According to the phenomena of osmosis, the solvent will move from low concentration of solute which is present in the egg to the higher concentration of solute which is present in this solution. So due to this the egg will get shrink. In osmosis, the solvent molecules move from the solution of low concentration of solute to the higher concentration of solute. Next is osmotic pressure. The flow of the solvent from its site to solution site, solvent site to solution site across the semi-permeable membrane can be stopped by some extra pressure applied on the solution site. Because osmosis is the process when the flow of solvent from its site to the solution site through a semi-permeable membrane, and this flow can be stopped by applying some extra pressure. So the excess pressure which must be applied to a solution to prevent the passage of solvent into it through a semi-permeable membrane is known as osmotic pressure. This can further be seen by the image also. We have two chambers. In one part solution is present and in other part solvent is present, and they both are separated by a semi-permeable membrane. And due to the process of osmosis, the solvent particle can pass through the solution through this semi-permeable membrane, but as we applied extra pressure on the solution side, because as it is an open container, so both the sides have a pressure which is known as atmospheric pressure. Atmospheric pressure will also applied on the side of solvent and also in the side of solution. But as we applied extra pressure on the side of solution side, means atmospheric pressure plus some extra pressure which is denoted by pi, then and it will prevent the flow of solvent to the solutions, then this pressure which is denoted by pi here is known as osmotic pressure. the solvent molecules will always flow from lower concentration to higher concentration of the solution. The osmotic pressure has been found to be depend on the concentration of the solution. Osmotic pressure is a colligative property as it depends on the number of solute molecules, not on their identity. For dilute solutions, it has been found experimentally that osmotic pressure is proportional to the molarity, that is concentration of the solution at a given temperature T. From this, osmotic pressure is denoted by pi, as we have already discussed. So from this, pi will be equal to osmotic pressure will be equal to concentration gas constant into temperature. Pi will be equal to CRT. Hence pi is osmotic pressure, R is gas constant, C is concentration. From this, since osmotic pressure is a colligative property and colligative properties are helpful in determination of molar mass of solute. Now from this we can also find out the molar mass of solute. Since concentration equal to number of moles of solute divided by volume, we all know that C will be equal to N2 upon V. On putting the value of concentration to the equation of osmotic pressure, we will get osmotic pressure pi equal to N2 into R into T upon volume. Since we know that number of moles of solute equal to mass of solute divided by molar mass of solute. From this, on putting the value of number of moles to the equation, we get pi equal to W2 into R into T divided by M2, which is molar mass of solute into volume of solution. From this, the value of M2 can be calculated. M2 will be equal to W2 mass of solute into gas constant into temperature divided by osmotic pressure into volume. Here is a question related to osmotic pressure. Calculate the osmotic pressure in pascals exerted by a solution prepared by dissolving 1 gram of polymer of molar mass 185,000 in 450 ml of water at 37 degrees Celsius. Given is molar mass of polymer, that is 1 gram. Molar mass of the solute, molar mass of the polymer is given, that is 185,000, and volume of solution 450 milliliter. From this, on converting the temperature into Kelvin unit, the temperature will be 310 Kelvin, and the value of gas constant R will in pascals, that will be equal to 8.314 into 10 to the power 3 pascal liter per mole per Kelvin. According to osmotic pressure, pi equal to W2 into R into T upon M2 into V. And here we have to find out the value of pi osmotic pressure. On putting all these value to this equation, we will get the value of value of osmotic pressure in pascal, and that is 30.96 pascal. Here we calculated the value easily. Here are some conditions for getting accurate value of molar mass. So first of all, the solute must be non-volatile in nature. The solute which we are adding, it must be non-volatile in nature. The solution must be dilute, that is concentration of the solution in the solution should not be more than 5%. It means the solution is diluted. The third condition for getting the accurate value of molar mass is the solute should not undergo either association or dissociation in the solution. Come to some biological significance of osmosis. The osmotic pressure method is widely used for measuring the molecular masses of proteins, polymers, and other macromolecules. Osmotic pressure is a method which is used for measuring the molecular masses of macromolecules like proteins and some bigger molecules like polymers. So for this, the solutions can be divided into three types. First is isotonic solution. Isotonic solutions are those in which the two solutions having same osmotic pressure at a given temperature are called isotonic solution. Next is hypertonic solution. If a solution has more osmotic pressure than the some other solution, then such type of solution are known as hypertonic solution. Third is hypotonic solutions. If a solution has less osmotic pressure than other solution, then it is called as hypotonic solution. Iso means same. When two solutions have same osmotic pressure, then they are known as isotonic solution. Hyper means higher. If one of the solution has higher osmotic pressure or more osmotic pressure than the other, then it is known as hypertonic. Hypotonic means when one of the solution has lower osmotic pressure than the other one, then that is known as hypotonic solution. For this, for isotonic solution, hypotonic solution and hypertonic solution, let us take an example. In the blood cell of human being, our blood cell contains a fluid, and that fluid has 0.9% of sodium chloride solution. Inside the blood cells, the fluid which is present in the concentration of 0.9% sodium chloride solution. As we put the blood cell in the solution containing 0.9% sodium chloride, as we put the blood cell in the solution containing 0.9% NaCl solution, then what will happen if we put, this is our blood cell, and if we put this blood cell to the solution containing 0.9% solution, then since the concentration of both blood cell and the solution in which the blood cell is put it are same and equal, so as the concentration is equal, then the pressure, osmotic pressure, will also be equal. And if two solutions having same osmotic pressure, then such type of solutions are known as isotonic solution. Come to the next point. If the salt concentration in which the blood cell is put it is less than 0.9%, we can see in example in image 2, if the concentration of solution, this concentration of solution is less than the concentration of the fluid which is present in blood cell, that is less than 0.9%, then what will happen at that time? The osmosis will occur, and osmosis is a process when the solvent particles will move from low concentration of solute to higher concentration of solute. It means here the solute particles of low concentration of solution, which is in low concentration in the solution, the solvent will move from this lower part of solute concentration to the higher part of the solute concentration, which is of blood cells, which is of 0.9%, which is present inside the blood cell. So what will happen at that time? At that time the blood cell will get swell. The blood cell will become a swollen. Why it is get swell? Because the due to osmosis, the flow of solvent molecule will takes place from low concentration of solute to higher concentration of solute, and higher concentration of solute in this case is present in blood cells. And due to low concentration of solution and higher concentration of blood cell, the osmotic pressure of the solution will also decreases, and such type of solutions are known as hypotonic solutions. Come to the third image, and in third image the concentration of the solution is higher, it is more than 0.9%. अगर ये जिस सॉल्यूशन में हम ब्लड सेल को पुट कर रहे हैं वो वही सॉल्यूशन ज़्यादा है 0.9% से, सपोज वो 1.2 1.3% सोडियम क्लोराइड सॉल्यूशन को कंटेन कर रहा है, इस कंडीशन में क्या हो जाएगा, जो ब्लड के अंदर फ्लूइड का कॉन्सेंट्रेशन था दैट इज 0.9% ओनली, और जिसमें हमने पुट करा वो 0.9% से ज़्यादा है, तो According to the process of osmosis, the flow of solvent molecule takes place from low concentration of solute to higher concentration of solute. तो low concentration तो blood cells के अंदर है, तो क्या होगा, जो low concentration blood cells के अंदर था वहां से solvent molecules बाहर को आने लगेंगे, कहाँ को, जहाँ को higher concentration है, क्योंकि हायर कॉन्सेंट्रेशन तो सॉल्यूशन में, तो सॉल्वेंट मॉलिक्यूल्स बाहर को आएंगे हायर कॉन्सेंट्रेशन ऑफ सॉल्यूट की साइड को, तो ये सारे के सारे बाहर आएंगे एंड ब्लड सेल विल गेट श्रिंक इन दिस कंडीशन, क्योंकि उसके जो सॉल्वेंट पार्टिकल थे वो कहाँ आ गए, सॉल्यूशन में आ गए, एंड ब्लड सेल विल गेट श्रिंक. एंड इफ द कॉन्सेंट्रेशन of the solution of one solution is higher than the other, then such type of solutions are known as hypertonic solution. Next is reverse osmosis. The direction of osmosis can be reversed if we apply large pressure larger than the osmotic pressure is applied on the solution side. Clear? That is, now the pure solvent flows out of the solution through a semi-permeable membrane, and this process is called reverse osmosis. If we applied a larger pressure on the solution side, larger than the atmospheric pressure on the larger than the osmotic pressure on the solution side, then what will happen? The solvent particles which is present on the solution, the solvent particles which is present on the solution will move towards the pure solvent side through a semi-permeable membrane, and this process is known as reverse osmosis. It is opposite of osmosis. In osmosis, movement of solvent particles towards the solution side takes place, but in reverse osmosis, on applying larger pressure on the solution side, the solvent particle will move towards there. The solvent particle from the solution will move towards the pure side through a semi-permeable membrane. This process is known as reverse osmosis, and reverse osmosis is used in desalination of seawater. Desalination of seawater, when pressure more than osmotic pressure is applied on salt water side. This can be seen by this image. This is in this image there are two chambers. One chamber is containing salt water and another chamber is containing fresh water. Salt water means it is a solution. It may contain many impurities. If we apply more pressure on salt water side, higher pressure than the atmospheric pressure on salt water side, then the solvent molecule which is present in this solution, the solvent molecule which is present in this solution will move through a semi-permeable membrane towards the fresh water side, and we will get fresh water from the water outlet. As we have to desaline the sea water, we apply the process of reverse osmosis. दो कंटेनर है हमारे पास डिफरेंट टाइप के, उनमें दो चैम्बर प्रेजेंट है. हमने एक चैम्बर में सी वाटर रखा जिसमें बहुत सारी इम्प्योरिटीज थी. दूसरा जो चैम्बर था उस चैम्बर में हमें प्योर वाटर आके मिलेगा. वो किस प्रोसेस के थ्रू मिलेगा? रिवर्स ऑस्मोसिस के प्रोसेस के थ्रू मिलेगा. आरओ जो सी वाटर था जिसमें बहुत सारी इम्प्योरिटी थी, हम उसके ऊपर प्रेशर अप्लाई कर रहे हैं और वो ऑस्मोटिक प्रेशर से भी ज़्यादा प्रेशर है. तो जो सॉल्वेंट के जो मॉलिक्यूल्स हैं और उन दोनों के बीच में जो हमने पार्टीशन करा हुआ एक सेमी परमिएबल मेम्ब्रेन प्रेजेंट है. तो जो सॉल्वेंट जो मॉलिक्यूल्स हैं वो बाहर निकल जाएंगे सेमी परमिएबल मेम्ब्रेन के थ्रू दूसरे चैम्बर में आके. सॉल्यूशन से बाहर निकल जाएंगे सॉल्वेंट मॉलिक्यूल. रिवर्स ऑफ ऑस्मोसिस इज देयर. इन ऑस्मोसिस सॉल्वेंट मॉलिक्यूल go to the solution side, but in reverse osmosis solvent molecules comes out from the solution side through a semi permeable membrane, and on doing desalination of seawater, after applying the pressure and as the solvent particles through a semi permeable membrane come to the another chamber, we will get fresh water from the water outlet. So this is the process of reverse osmosis. And that's all for today. I have some questions for you. Try to solve this question and we will discuss our next topic in our next video. There are some practice questions for you. Try to solve these questions. Thank you. That's all for today.